Bonjour et bienvenue à l'allocution d'ouverture en sciences fondamentales. Je suis heureuse, je suis ravie d'être capable d'introduire notre conférencière aujourd'hui, Dr. Xu Yu. Elle est membre de l'équipe principale de l'Institut Reagan, de l'Hôpital général du Massachusetts, de l'Institut technique du Massachusetts et de l'Université Harvard. Elle est professeure agrégée en médecine à l'Hôpital général du Massachusetts, à l'Hôpital Brigham and Women's et à la Faculté de médecine de Harvard. Elle est membre élue de la Société américaine en sciences cliniques, en recherche clinique. Son laboratoire se concentre sur la connaissance des mécanismes moléculaires et cellulaires en cause dans le contrôle immunitaire de l'infection virale. Ses travaux récents sur les profils des réservoirs viraux chez les contrôleurs élites du VIH ont été choisis comme l'une des 9 candidatures en première de l'année 2020 dans la revue Science. Le titre de sa présentation sera « Contrôleurs élites du VIH comme modèle pour la guérison du VIH ».
Good morning, everyone. It is my pleasure to welcome you all to the Basic Sciences Keynote for CAHR 2022. We are honored today to welcome Dr. Xu Yu as our guest speaker for this session. Dr. Xu Yu is a core member of the Reagan Institute of the MDH, MIT, and Harvard, and an associate professor of medicine at Massachusetts General Hospital, Brigham and Women's Hospital, and Harvard Medical School. She's an elected member of the American Society for Clinical Investigation. Her laboratory focuses on understanding molecular and cellular mechanisms involved in the immune control of viral infection. Her recent work on viral reservoir profiles in HIV elite controllers was selected as one of the 9 runners-up to the 2020 Breakthrough of the Year by Science magazine. The title of her presentation today is HIV-1 Elite Controllers as a Model for HIV Cure. Dr. Xu Yu, it is our honor to have you here. Please take it away. Thank you.
Okay, yeah, thank you so much for the introduction. Can you guys hear me okay?
Yes.
Great. Yeah, many thanks to the organizers also for the invitation. In this presentation, we will review what we learned from HIV elite controllers and then discuss how they might be relevant to the rest of the people living with HIV. So we know that HIV infection is an incurable disease, mainly because HIV infection forms cellular reservoirs that can persist for lifelong, even in the setting of suppressive antiretroviral therapy. And the number of these viral reservoir cells is very small, typically in the range of 1 to 10 cells per million CD4 T cells, but they can fuel HIV rebound viremia very quickly after treatment interruption. So when it comes to a cure of HIV infection, we usually discuss 2 forms of possible cure. A more classical form of cure is achieved when all HIV infected cells, or at least all HIV infected cells that encode for replication-competent HIV are eliminated. And the best example for this form of HIV cure is the story of Mr. Timothy Brown, the Berlin patient. However, a long-term drug-free remission from HIV infection may actually be possible even in persons with persisting reservoirs of HIV. Replication-competent HIV proviruses are readily detectable in these individuals. However, these people are still able to maintain undetectable plasma viral loads in the absence of therapy. So typical examples for durable remission of HIV infection are natural HIV ED controllers and some post-treatment ED controllers. As you know, natural HIV ED controllers represent a very small proportion of all HIV infected individuals. but at the same time, they are probably the best model for studying a cure of HIV infection. In order to systematically study these elite controllers, the International HIV Controller Consortium was established about 15 years ago and has recruited hundreds of HIV elite controllers so far. So generally speaking, HIV elite controllers are HIV-infected individuals with at least 3 consecutive undetectable plasma viral load measurements over at least 1 year without any antiretroviral treatment. This unique cohort really gives us the opportunity to look into 2 major questions. First, can a cure of HIV infection occur during natural infection? And what are the characteristics of a drug-free, durable remission of HIV infection? So another way to ask these questions would be, would it be possible that some elite controllers no longer have any replication-competent reservoirs, which means they have achieved a cure of HIV infection on their own? And if there are still replication-competent reservoirs in some other elite controllers, what would be possible distinguishing features of their HIV reservoir cells relative to the majority of the people living with HIV who actually need continuous antiretroviral treatment to achieve viral suppression? To address these questions, we recruited 73 elite controllers who have been infected with HIV for a median of 18 years. Over 60% of our elite controllers had no detectable viremia throughout their clinical follow-ups for a median of 9 years without needing any antiretroviral treatment. And for the remaining 38% of the elite controllers with some viral blips, they typically only had 1 blip during the follow-ups with less than 100 copies of HIV RNA detected per ml of plasma. So as a comparison group, we recruited 41 ART-suppressed HIV-infected individuals who similarly had a median of 9 years of undetectable viremia, but obviously through continuous suppressive antiretroviral treatment. As we know, HIV is a retrovirus. It has the ability to integrate its own genetic material into the human genomic DNA in target cells to form the cellular reservoirs. So when discussing HIV reservoir cells, it is very important to differentiate two types of HIV proviruses, the intact and defective proviruses. The majority of integrated HIV proviruses are defective. They can be transcribed, but cannot lead to replication-competent viruses. And these defective proviruses frequently account for about roughly 90% of the proviruses detectable in a given HIV-infected individual. So basically, these defective proviruses are like fossils indicating the past history of HIV infection of these cells. In contrast, if the proviruses are intact, they can then behave like a regular human gene to generate new viral RNAs and proteins and give rise to many new replication-competent viruses when the opportunity comes. And these proviruses can then be carried on to all the daughter cells from the same infected cells during the clonal expansion and could in this way persist for lifelong. But the intact proviruses only represent a minority of all provirus sequences in the reservoir. So over the past few years, several groups have developed near full-length provirus sequencing assays, which allow to interrogate the proviral reservoir at the level of single proviral genomes. So this assay can distinguish intact, potentially replication-competent proviruses from the majority of the proviruses that are defective. So using this full genome sequencing assay, we were able to quantify the proviral reservoirs in the 2 study cohorts. So as expected, we find significantly lower levels of both total and intact proviruses in elite controllers compared to ART-suppressed individuals. But surprisingly, we observed more profound variations in the frequency of intact proviruses in elite controllers. Some elite controllers has very high levels of intact proviruses, higher than most of the ART-suppressed individuals, while others have extremely low to undetectable levels of intact proviruses. So especially there are 2 elite controllers who had no detectable intact proviruses despite analyzing large amounts of cells. And the question here is whether a cure of HIV infection may have occurred naturally in these 2 exceptional elite controllers. As we know that the Berlin patient and more recently the Linden patient have been the gold standard for a cure of HIV infection. So both of them received stem cell transplant from donors with homozygous CCR5 delta 32 mutations as the treatment for their blood cancer, which also helped them to cure HIV infection at the same time. So extensive virological testing was done for the Berlin patients. As highlighted here, despite analyzing 1.4 billion CD4 T cells in viral outgrowth assays, no replication-competent viruses were detected. A more limited number of cells were analyzed in the Linden patient. Some HIV DNA were detected in memory CD4 T cells. However, it was not clear if these proviruses were intact or defective viral DNA. When HIV DNA from tissues were analyzed, small amounts of viral DNA were detected in the rectal biopsy samples from the Berlin patient and in lymph node samples from the Linden patient. Again, it is unclear if these were genome intact proviral DNA or defective viral DNA, although I think most of us would agree that these are most likely represent only defective proviruses. So the first exceptional elite controller we studied in great details is Ms. Lauren Willenberg, who was diagnosed with HIV infection 30 years ago, had never been treated, and had undetectable viremia for the past 27 years that we have in the record, except one viral load test of 93 HIV RNA copies per ml of plasma in early 2000. So she was initially referred to as the San Francisco patient. In her case, we failed to detect intact or replication-competent proviruses despite analyzing more than 1.5 billion blood cells using the full genome sequencing assay and more than 340 million resting CD4 cells in the viral outgrowth assay. So more recently, we have identified a second exceptional elite controller. This person was diagnosed with HIV infection in 2013, and all the viral tests over the past 9 years were below detection threshold. No ART was started until 2019 during the second and third trimesters of her pregnancy. After delivering a healthy HIV negative baby, she stopped ART and her HIV viral loads remained undetectable. Similarly, this person has no sign of intact proviruses after analyzing 1.2 billion blood cells and 500 million mononuclear cells in the placenta using the full genome sequencing assay. And we also did not detect any replication-competent proviruses out of 150 million resting CD4 T cells using the viral outgrowth assay. So this person is originally from the city of Esperanza in Argentina, and in line with her wish, we named her the Esperanza patient to send a message of hope for a cure of HIV infection. However, in both of these 2 exceptional controllers, we did detect multiple defective proviruses in their peripheral blood cells. In the San Francisco patient, we detected 4 near full-length HIV DNA sequences with ApoBax3G and 3F induced hypermutations, and additional 15 HIV proviruses with large deletions. Interestingly, it seems that the reservoir cells harboring HIV DNA sequences with this large deletion had clonally expanded and persisted for at least 10 years. So in Esperanza patient, a total of 7 defective proviruses were detected, including 1 near full-length HIV DNA sequence with hypermutation, and 6 additional provirus sequences with large lesions. Again, the reservoir cells harboring this HIV DNA sequences which large deletion had clonally expanded and persisted for at least 2 years. These defective HIV DNA products clearly indicate that both exceptional elite controllers were infected with HIV, and some active cycles of viral replication had occurred in the past in vivo in these 2 patients. If we look more closely into the HIV DNA sequences with large deletions, we find that these sequences differ at multiple base pair residues. And this further suggests that this is not simply an abortive infection of the founder virus, but instead multiple rounds of productive infection have occurred in the past. In addition, as you know, that the NAF deletion have been previously associated with elite control of HIV, but the near full-length hypermutated sequences in both of these exceptional elite controllers do not have NAF deletions. Therefore, we believe infection with an attenuated virus strain is unlikely the cause of elite control of HIV infection in these 2 individuals. In addition, the Esperanza patients, we actually typed it, and she has the homozygous Y-type CCR5 gene, and her activated CD4 T cells express clearly detectable levels of both CCR5 and CXCR4 receptors. And her cells were able to effectively support HIV replication in vitro infection assays with both R5 and X4-tropic viral isolate. So overall, these data indicate that similarly to the Berlin patient, these 2 exceptional elite controllers we identified also have no detectable intact or replication-competent HIV proviruses in massive number of cells that were analyzed using several complementary assays. Although it can never be definitively proven, we believe that these 2 exceptional elite controllers probably have achieved a cure of HIV infection through their natural immunity. So now I'm going to switch to the elite controllers with a higher level of intact proviruses, despite undetectable plasma viremia for decades. So these elite controllers fulfill the definition of a durable drug-free remission of HIV infection, but they did not develop a cure of HIV infection. So this raises the question of how these elite controllers can still maintain spontaneous control of HIV despite these large intact proviral reservoirs. To address this, we focused on the chromosomal locations of integrated HIV proviruses. So over the past 2 decades, several different assays has been developed to map HIV integration sites within the human genome. Using these assays, prior studies have consistently shown that in both CD4 T cells from HIV-infected individual or in vitro infected CD4 T cells, about 80 to 90% of HIV proviruses were integrated into the genes, which usually only make up a small fraction of the human genome. So in contrast, 10 to 20% of the HIV proviruses were integrated into non-genic regions that constitute the majority of the human genome. So there are clearly preferential integrations of HIV proviruses into the genic regions of the human chromosome. However, these previous analysis did not allow to distinguish the chromosomal integration sites of intact versus defective proviruses. To specifically address this technical issue, we developed a new analysis pipeline, which we call the Matched Integration Site and Proviral Sequencing Assay, or MAPE-seq. So briefly, after diluting the proviruses to single genome levels, we conducted a whole genome amplification step, which gives us enough material to do both near full-length proviral sequencing assay as well as the corresponding chromosomal integration site for individual proviruses. So here's an example of integration site profile of intact proviruses in an HIV infected but ART treated individual who actually has been on suppressive ART for 11.5 years. So this is a phylogenetic tree of all intact proviruses we detected in this individual. Each branch on the tree indicates one intact provirus sequence, and the black dots indicate each of these sequences have been detected more than once. The pink colors indicate the integration sites in the genic locations, while the blue letters are for the integration sites in non-genic locations. So as you can see on this phylogenetic tree, the intact proviruses in this ART-treated individual are quite diverse, with the majority integrated into the genic regions, and only a small proportion of intact proviruses are in the non-genic or pseudogenic regions. In contrast, here's an example of an HIV ED controller who was diagnosed with HIV infection 28 years ago, have never been treated, and had no detectable viremia, at least for the past 12 years that we have record for. And you can see that while doing very well clinically, this ED controller has one of the highest intact proviral reservoir sizes in our ED controller cohort. It is actually higher than most of the ART suppressed persons. So here again is the phylogenetic tree of all intact proviruses detected in this ED controller. Each symbol on the tree is an intact HIV sequence. The black box around the sequences indicate clones with 100% sequence identity. So unlike the diverse profile we just saw in the ART-treated individual, in this ED controller, we have only detected 2 large clusters of clonally-expanded intact proviruses. So one of these intact proviral clone is integrated into Centromeric satellite DNA region in chromosome 7. As you may know, the Centromeric satellite DNA is a form of Heterochromatin and is far away from any host transcriptional start site. And therefore, it's also referred to as a gene desert. So intact HIV proviruses integrated into these regions would have very limited chance to host gene transcriptional machinery that most likely are in a state of deep viral latency, which means they would have very limited chance to be reactivated or to produce replication-competent viral particles in vivo. So the second clone of intact proviruses is integrated into a KRAB-ZNF gene in chromosome 19, which is also characterized as a heterochromatin. So this region is usually packed with repressive chromatin marks. Again, intact proviruses integrated into these regions are also likely in a state of deep viral latency. So to summarize, we have obtained chromosome integration sites from 103 and 113 intact proviruses from 15 ED controllers and 7 ART-suppressed individuals. In ART-suppressed individuals, you can see that the majority of intact proviruses were integrated into the ordinary genic regions, which are more permissive to viral reactivation. However, over 60% of the intact proviruses in ED controllers were integrated into non-genic regions, satellite DNA regions, or the GNF gene in chromosome 19. So all of which are associated with deep viral latency. So this really raises the next set of questions. Is the intact proviral integration landscape in ED controllers related to altered integration site preferences at the beginning of infection, or is it the result of immune-mediated clearance of intact proviruses located in the permissive chromosomal locations over time? We addressed these questions in 2 ways. So first, we infected CD4 T cells from 12 HIV ED controllers and 9 HIV negative healthy individuals with the GFP encoding HIV construct. We then sorted GFP positive and GFP negative CD4 T cells and then conducted chromosomal integration site analysis. So these experiments retrieved over 120,000 HIV integration sites. So overall, these data demonstrate that there was no evidence of preferential targeting of centromere satellite DNAs or KRAB-ZNF genes in chromosome 19 in these in vitro infected CD4 T cells from controllers. They were very similar to what you see in the CD4 T cells from HIV negative donors. In addition, we obtained integration site on 45 and 84 defective proviruses in the same 15 ED controllers and 7 ART suppressed individuals. So the integration of defective proviruses in ED controllers really resembles what you see in ART suppressed individuals, with the majority located in the regular genic regions. So this indicates that the proviral integration by itself is really not biased towards non-genic or heterochromatin regions in ED controllers. So just to quickly summarize, we believe both sets of experiments indicate that the integration site profile of intact proviruses in ED controller is most likely the result of immune selection pressure that was able to effectively eliminate intact proviruses in more permissive genic regions. And it's also very interesting to note that the intact and defective proviruses seem to be under different levels of immune selection pressure. Apparently, the immune system is able to mount more pressure on the intact proviruses in accessible chromatin, and resulting in a more pronounced accumulation of intact proviruses in the heterochromatin regions. But this is not true for the defective proviruses. So we've shown that HIV integration sites within this non-genic or heterochromatin regions are associated with spontaneous control of HIV infection. But one remaining question is whether these particular proviruses are truly more transcriptionally silent when it comes to the viral gene expression. So in order to answer this question, we have developed a third generation assay called PRIP-seq. So after diluting HIV reservoir cells to single cell level, DNA and RNA are separated, and the genomic DNA is then subjected to the whole genome amplification for matched proviral sequencing and integration site analysis, very similar to what we described in the MIP-seq assays, while the RNA is reverse transcribed and amplified for HIV RNA transcriptional analysis. Therefore, this PRIP-seq assay allows the parallel identification of proviral sequence, chromosomal integration site, and HIV transcriptional activity for each single viral reservoir cell without even needing any ex vivo stimulation. We have done the parallel analysis of these three components in 701 reservoir cells from 6 ART treated individuals. Very consistent with the prior study, about 17% of the proviruses were integrated into non-genic regions. So when we look at the transcriptional activities of these proviruses, we can see that the proviruses in genes are quite transcriptionally active. About 30 to 40% of these proviruses in genes are producing some viral RNAs, while a significantly lower proportion of the proviruses in the non-genic regions were transcriptionally active. So when we're looking specifically at the proviruses integrated into non-genic satellite DNA regions, we observed that only 2 out of 34 proviruses in these regions were transcribing any detectable levels of HIV RNA. So as mentioned earlier, these are the very regions in which intact proviruses from ED controllers are preferentially located. It seems that they are associated with a deep viral latency in this direct ex vivo analysis. So overall, these data really indicate that location matters. When an intact provirus is integrated into an accessible chromatin, it is more likely to be actively transcribed, which may allow the intact provirus to give rise to replication-competent viruses. However, if an intact provirus is integrated into non-genic or tightly packed heterochromatin regions, this intact provirus cannot be easily reactivated and is likely in a deeper state of latency. We refer to this state as blocked and locked status. So here's what we propose is happening in these ED controllers. So after early infection, most of the intact proviruses were located in the accessible chromatin where they can be readily expressed, but only very few intact proviruses were integrated into non-genic or heterochromatin regions with deeper latency. When there's an autologous reactivation signal, such as cytokine or antigen stimulation, intact proviruses in accessible chromatin have a high likelihood to be transcribed, which will then expose the reservoir cells to the host immune system. This will increase the probability that they are eliminated by the various host immune defense mechanism. So we refer to this process as the autologous shock and kill process. We propose that in naturally cured patients, through the autologous shock and kill process over time, all intact reservoirs were completely cleared by their natural immunity. But in the majority of the ED controllers, we hypothesize that the intact proviruses in accessible chromatin are gradually eliminated over time, while the intact proviruses in deeper latency persist and even expand. So eventually, the remaining intact reservoir may be largely confined to the heterochromatin positions where HIV is blocked and locked and cannot be easily reactivated. So it appears that such a blocked and locked intact proviral reservoir profile enables a drug-free and a durable remission of HIV infection in our ED controllers. So now the big question is, how can we apply what we learned from ED controllers to the general population of people living with HIV? Would our autologous shock and kill process also drive the evolution of the proviral landscape during long-term antiretroviral therapy? Even though we can imagine this process is probably much slower and much less effective in ART-treated individuals. And would it be possible that some of the long-term ART-treated individual may have already had a blocked and locked intact proviral reservoir profile that would allow them to achieve durable drug-free remission after treatment interruption? Or in other words, they may become post-treatment controllers who do not need continuous antiretroviral therapy anymore. So in order to address this question, we specially established a new cohort, the long-term ART cohort, which include 8 study participants who have been on suppressive ART for a median of 20 years. So these are the exact population who started treatment since ART was first introduced. For some of these study participants, there were historical samples soon after treatment initiation and around 10 years after continuous treatment, as well as a recent sample after about two decades of ART treatment. So this figure on the right is really a good example of the study participants in our long-term ART cohort and the sampling time point that we included for the longitudinal study on this cohort. We also included 2 existing cohorts as the reference cohorts. So one has 43 participants who have been on suppressive ART for a median of 9 years, which we call the moderate ART cohort. And the other one is our ED controller cohort with 72 participants who can maintain undetectable viremia without any treatment. So first, we compared just cross-sectionally the levels of total and intact reservoir size in these long-term ART-treated individuals after 20 years of ART, and compare these with the reservoir size in the 2 reference cohort. We saw no significant difference in the quantity of total and intact HIV reservoirs between the long-term ART and the moderate ART cohorts. But as expected, both ART cohorts have higher total and intact reservoirs than the elite controllers. However, when we look at the proportions of intact proviruses within the total reservoirs, we started to notice a difference. The proportion of intact HIV proviruses in long-term ART participant is similar to that in the elite controllers, but significantly higher than in the moderate ART. And this is mainly due to the fact that a higher proportion of clonally-expanded intact HIV reservoirs are found in both long-term ART and elite controllers, and much lower in the moderate ART cohort. So I just want to show you some examples of the intact proviral reservoirs in our long-term ART cohort after about 2 decades of ART treatment. So in these 2 individuals, we see almost monoclonal intact reservoirs enriched in the centromeric or pericentromeric satellite DNA regions, which probably reminds you of what we have seen previously in elite controllers. And here are another 2 individuals in our long-term ART cohort. Even though these 2 participants have more diverse intact reservoirs with multiple clones, what's interesting is that almost all these intact clones are in non-genic or heterochromatin regions that are associated with deeper latency. So in summary, the intact proviral integration site profile in these long-term ART cohort is strikingly similar to that in elite controllers, with very high proportions of intact proviral integration sites in centromeric DNA regions or non-genic regions or GNF gene regions, and all the sites that are associated with deeper latency. So now we have characterized the intact reservoir profile after these prolonged periods of ART. We want to determine how these reservoirs have evolved or changed over time. To answer this question, we analyzed reservoir profile of intact proviruses from the earlier time point, as I showed you before, for these long-term ART study participants. So I just want to walk you through this circus plot. So each of these circus plot, each of these bars represent one intact proviral sequence, and each of these colorful arch indicates a clone of intact proviruses with 100% sequence identity. So the earliest time point we analyzed for this individual is 1 year after ART initiation. So we detected several individual intact proviruses and a large clone of intact proviruses. As you can see, all of the intact proviruses detected at this early time point are in genic regions. So in the second time point, after 12 years of suppressive ART treatment, we see the integral clones still persisted, but much smaller. And there was another small clone in genic region were detected. But at the same time, we detected a new large clone of intact proviruses in centromeric DNA region of chromosome 18. So in the latest time point, after 20 years of suppressive ART, we see that the intact provirus integrated into this centromeric regions of chromosome 18 comes to dominate the intact reservoir, resulting in a nearly monoclonal intact reservoir profile in this individual at this time. Here's another example in this individual. After 1 year of ART initiation, the majority of integration sites of intact proviruses are in genic and permissive regions. But interestingly, we have already detected some intact proviral sequences in non-genic or GNF gene areas at this very early time point. So especially we have one clone that is in microsatellite DNA region in chromosome 10. After 9 years of ART treatment, the chromosome 10 pericentromeric clone persisted, and we also see a new chromosome Y pericentromeric clone appeared. And we also detected some individual intact proviral sequences in non-genic or GNF gene regions. So the previously detected intact proviruses in the genic regions were not detectable anymore. In the final time point, after 20 years of ART, the original pericentromeric clone on chromosome 10 still persisted, so lasting for 2 decades by now. The newly detected chromosome Y pericentromeric clone in the second time point expanded greatly and dominating the intact reservoirs in this late time point. So collectively, from these 2 examples, what the longitudinal analysis really showed us is that over time, we see a progressive accumulation of intact proviruses with integration site features of deeper latency. In contrast, proviruses in genic positions frequently detected only in the early time points are selected against over time and were rarely detected at later time point after prolonged ART. So the previous 2 surface plots are for the intact proviruses, but when we look at the defective proviruses from these participants, we actually did not detect any selection, did not see any evidence for selection of defective proviruses in deeper latency over time. And we could easily still detect defective proviruses in genic regions even after 20 years of suppressive ART. So this again supports our previous observation that the intact and defective proviruses seem to be under different levels of immune selection pressure in vivo. So to summarize these longitudinal proviral profile analysis, from the first time point to the last time point over 2 decades, we see a significant increase of intact proviruses found integrated into centromeric, non-genic, or ZNF genes, all the sites that we talk about that are associated with deep viral latency, while this trend does not exist for defective proviruses over time. So we believe that what's happening in these individuals during long-term ART is very similar to what's naturally occurring in elite controllers. The intact proviral reservoir profiles we see after 2 decades of ART is due to an immune selection process that specifically eliminates intact proviruses in more permissive chromatin locations, while the intact proviruses in heterochromatin regions are accumulated over time. It is possible that the blocked and locked intact proviral reservoir profile after long-term ART may lead to post-treatment control or drug-free HIV remission after treatment interruption in these individuals. However, this really needs to be evaluated extensively in future clinical trials. Another critical question is how this autologous shock and kill process could be accelerated through therapeutic interventions during antiretroviral therapy, so that more HIV-infected individuals could reach a blocked and locked reservoir profile after hopefully only a limited duration on suppressive ART. Obviously, for this, we need to identify the host immune factors that drive the progressive immune selection of intact proviruses in deeper latency in elite controllers, as well as in these long-term ART-treated individuals. But I believe that this is an area that many investigator teams are actively working on. So just to quickly summarize, a cure of HIV infection may represent a rare but possible outcome of HIV infection once all replication-competent or genome-intact proviruses are eliminated through natural immunity. And drug-free and durable remission of HIV infection could possibly be achieved in the HIV-infected individual once all intact proviruses are in the blocked and locked positions in the host genome, like the reservoir profile that we see in elite controllers. Our data also suggests that not the frequency of intact proviruses by itself, but instead their chromosomal locations may be an important distinguishing features for a durable remission of HIV infection. Therefore, we believe that assays able to capture qualitative reservoir profiles will be needed to evaluate effects of future immunotherapeutic interventions aiming for durable remission of HIV infection. Finally, I would like to thank all group members in my lab and each Felt's lab, as well as all our long-term national and international collaborators. Most importantly, none of these studies would be possible without the dedication of all our study participants to the biomedical research, which we are always extremely grateful for. And also many thanks to our funding agencies. And thank you for your attention. I'll be happy to answer any questions that you may have.
Thank you so much. That was so interesting. I see that there are already several questions in the chat. So maybe I'll start. I think there are 3 questions yeah, I'll start with the one where 3 people have indicated that they want to comment on this. Do you see more viral blips in elite controllers as they age? And does this immune aging affect the ability of elite controllers to control their infection?
Yeah, this is a really interesting question. I don't think we have any data to support one conclusion or the other, but this is definitely a very interesting area to study, especially with the HIV infected population getting aging more and more over the next few years. We do believe that the age would impact the epigenetic features of some of the locations these intact proviruses located. So this could have an effect in terms of viral blip in the later years, but we have not studied this very extensively. But that's definitely an area of research that we're interested.
Jim, you want to ask the next question?
Sure. I guess there's a question from the audience that asks if pregnancy itself would be perhaps associated with viral blips in elite controllers. Do you know anything about that, perhaps?
Yeah, sorry, we actually don't know that. I don't think we have studied any. Yeah, we don't know the details of the pregnancy status of our elite controllers in the staff cohort.
Okay, Nicole, can you get the next one?
Sure. Does sex play a role in being an elite controller? Are these elite controllers more likely to be male or female?
Yeah, very interesting. So this is 2 interesting observations. So as in our elite controller cohort, is still male dominating the cohort as any other HIV infected cohort, but doesn't mean we can conclude that more male will be have a chance to be elite controllers. And what is really interesting is the observation the 2 possibly naturally cured patients are both females. So we are hoping to expand this cohort to see whether we see any gender bias in terms of the ability of the immune system to clear intact proviruses in vivo. That's definitely a question, but obviously a beautiful cohort would help us to answer this question.
Another question is, have you done any work or plan to do any work looking at what immune pressures, for example, HLA, may be influencing provirus integration into non-genic sites?
Yeah, we're definitely interested. As I mentioned, probably many groups are working very actively to find the magic immune responses in these elite controllers. But from the data we have, I don't see we see a bias of particular HLA class 1 alleles. We know there's a slightly enrichment of B27 and B57, the so-called protective allele in our elite controller cohort. But in terms of reservoir profile, we have seen in these patients, we do not see a particular bias towards any particular HLA type. So apparently could be not only T cells, but other immune factors also contribute to the clearance of intact proviruses in the accessible chromatids.
Have you, in the last part that you were showing us with the long-term treated individuals, how do you have any feeling for how how often this happens? I mean, and you showed us data for 2 people, but does it happen more frequently, like for more people? I mean, the idea has been that you have to treat basically for a lifetime in order to cure HIV, but what this is telling us is maybe you don't. I mean, 20 years is a long time, but still it's better than a lifetime. So how generalizable is the phenomenon?
Yeah, no, this is a great question. Actually, the study looked at 8 patients. So in these 8 patients, we all see after about 20 years have this blocked and locked reservoir profile. And obviously, everybody is different. From the 2 patients I've shown you, you see somebody not fully cleared until 12 years, but the other person was in a really good shape even after 9 years. So clearly, this is a not very rare phenomena, but just how fast, how effective it could be, that probably depends on different people, how their immune system is functional over this long period of time. So this is definitely a very active area of research that we are conducting. We're collaborating with many national clinical sites to try to recruit long-term ART treated individual. So now we are hoping not only look at 20 years, probably 15 years, to see how many of these individuals have already achieved this kind of a blocked and locked reservoir profile. And the ideal scenario would be if we identify, if not everybody, but at least a significant proportion of these long-term ART treated individuals, we could possibly have a supervised treatment interruption studies to see whether it's possible they will have delayed viral rebound or even become completely post-treatment controllers that do not need antiretroviral anymore, like the elite controllers. Yeah, but as I mentioned, this is really need a well-designed, closely followed clinical trial to do this properly.
Okay, we have a question from Lyle. Lyle says, amazing talk. Given the accumulation of intact HIV and gene deserts happens naturally over time, either naturally or with treatments, why have so many therapeutic vaccine trials failed? Have they not been carried out for long enough or not induced the right type of responses?
Yeah, I mean, obviously, I think this is also, that's what I just mentioned. We need to profile the reservoir first on the patient before we stop treatment on anybody. That's our opinion. Many of the prior studies, probably the cohort is very diverse, and many of them actually are relatively short-term ART. So many of the intact proviruses are still in the accessible chromatin that's very easily reactivated once the treatment is interrupted. So we didn't, definitely the timing is a problem. We didn't give long enough duration for any therapeutic interventions to kick in and to really eliminate all the intact reservoir in the accessible chromatin. So that's why we believe if the therapeutic vaccine can induce a strong immune responses, probably not right after the vaccination to stop treatment, maybe give some time, allow the immune responses to act against or select the reservoir profiles more towards to the blocked and locked reservoir before treatment interruption studies to evaluate the vaccine efficacy in that way. Yeah.
Do different subtypes of HIV have preferential integration sites? And if so, would it explain how some subtypes can be more pathogenic than others?
This is a great question. I don't think we have enough study or data on different cohorts. We do have some samples from South African or Botswana. In that sense, we do see some also after a little bit long-term ART, we see accumulation of intact proviruses in these heterochromatin regions as well. So we don't have any hint at this point that there is a difference in terms of viral integration or selection forces of integration sites of intact proviruses in different clade or geographic locations.
Okay. We have a question from Amit. When you analyze provirus from patients that are on ART for different lengths of time, say 1 year versus 20 years, have you noticed any differences in ApoBec footprint, or are their VIFs any different?
Yeah, no, we have have the data. I don't think we analyzed that particularly. We can definitely go back to look at it. Or anybody want to collaborate, you can reach out to us as well.
Does deeper latent HIV integrate into resting memory T cells or other T cell types, or other cell types, excuse me?
Yeah, so for our analysis, we basically either look at the total PBMC and we want to capture all the reservoirs, possible reservoir cells, or look at the total CD4 or memory CD4. But I don't think we can differentiate at this point in which exact cell phenotype it is. And obviously that need really technology able to capture individual cell and individual sequence intactness and the location at the same time. Yeah.
So perhaps I could ask another question. There are ECs are heterogeneous, and there are some people that lose control. But in the model that you're proposing, you should be kind of going towards continued control rather than losing control in these elite controllers. So can you make a comment or maybe speculate as to what's going on in the elite controllers that lose control?
Yeah, no, that's a really interesting question. So as I mentioned, our cohort was quite selective. We really look at the patient have very long history of infection, has really no very blip for a prolonged period of time. So this is a perfect model for cure that's in our understanding. So that's why their reservoir profile really just tell us what the possible cure could look like. But the patient who lost control, it is well possible those patients still have some of the intact proviruses in accessible chromatin, but not reactivated at the earlier time point. But as we mentioned, with aging, with other immune stimulus, and some of the reservoir reactivated and cannot be controlled by the host immune system anymore, then that leads to a rebound. And obviously, all the study currently we're doing is in the peripheral blood. So we haven't really analyzed the tissue reservoir. So whether the reservoir in the profile in the peripheral blood mirrors exactly what's going on in tissue, that's another question. So possibly the rebound could come from the tissue reservoir that have not been studied very well at this point.
So are there any more questions?
I think we're good.
Yeah, I think that we've gone through all of the questions. So thank you so much. This was just an amazing presentation of also a lot of new work that you've been doing, which was great to hear about.
Great. Thank you. Enjoy the rest of the conference, everyone.
Thank you very much.
Bye.
Thank you.
Bye-bye.